Welcome everyone. Here we are at week number 5. So we are on the downhill slope of this term for Biology 120 Lab. Let's see what we've got going on. Wait a minute, gotta find my arrows. Okay, week 5. So last week you learned about cellular respiration. You did an experiment with the peas to see how much oxygen was taken in by the germinating peas. Hopefully that's what you got. And cellular respiration is the process where food is broken down to make ATP molecules that run all the work of enzymes, but it's done in the presence of oxygen. Oxygen helps this process be carried out, and so that's why you are measuring oxygen consumption in that cellular respiration experiment with the peas. This week our topic is DNA. DNA, I love me some DNA. Here's DNA. Everybody knows, wait a minute, there you go. Everybody knows what the molecule looks like. You've all seen it before. Double helix. Has the 4 bases, A, T, G, C, adenine, thymine, guanine, cytosine, that pair up across that double helix. And because of that pairing, it is very easy for DNA to be replicated or to be copied over and over again, and also to be used to make proteins. So the job of DNA is to contain the code to make all the proteins, and most of those proteins are enzymes that carry out biochemistry, to make all the proteins that an organism needs. And so all those proteins determine what kind of organism you are, because it determines what kind of biochemistry is taking place in your cells. So that's the importance of DNA. We're going to talk about copying it through a process called PCR, which you've heard of before by now. And you're going to isolate some DNA from your cheek cells. Okay, so let's get started. Milestone 2, your experimental design for your project in this course is due this week. So the experimental design is you are writing down your procedure for how you're going to do this thing that you've decided on, okay? Whether it's air pollution or the nervous experiment or the Manduka caterpillars or the ice experiment, whatever it is that you've decided to do. First of all, fix milestone 1. You'll have some comments in there from me so that you can fix what you need to fix in milestone 1. Make sure you use the correct format for milestone 2. This is a Word document, 1 to 2 pages long, and use bulleted lists. So I don't want to see full paragraphs. Do this. I'm going to do that. No. Bulleted items, step by step. So use the correct format. Include all the steps that you're going to take in your experiment. Okay, you've got to think through this experiment and design it so that then you're able to do it. All right, so the labs that you're doing this week, lab number 5, the cheek cell DNA extraction experiment, won't take you that long. It's very easy. I think it's very easy. There are a couple of things that I want to mention though, and I'm showing you in my pictures right here. That's my DNA. So you see this sort of slimy stuff in the middle of the tubes? That's the DNA, and that's all you're going to be able to see. I mean, that's a lot of DNA in order for you to see it. When we look at DNA, when we look at the variable regions in DNA to see differences between us and figure out our ancestry and determine who left behind evidence at the scene of a crime and all that sort of thing, you're looking down on a molecular level. Now, we are not looking at that with this experiment, but you will see your DNA. A couple of hints about this lab. CC stands for cubic centimeter. A cubic centimeter is a milliliter. Okay, just realize those are equivalent measurements. So realize that when you're using that cup. Your ethanol, the alcohol in this case, has to be extremely cold in order for you to isolate your DNA. And that does not mean from your refrigerator, and that does not mean putting it in your refrigerator for an hour before you use it. Put your ethanol in your freezer right now as you're watching this. Just pause it and go put it in the freezer until you're ready to do the experiment. And only take it out at the time that you need it, okay? Because it needs to be extremely cold for this to work. And if it is, it will work. This is a pretty quick lab. I know the instructions say that it's going to take about an hour. I don't really think it's going to take you an hour. It's a pretty quick lab. Then you're going to do the PCR simulation and answer the questions. And I'm going to put a link to a different PCR simulation in this announcement so that you can also go to it. I didn't really like the one that they have. It's okay. It's okay. But you're watching somebody else click through the simulation. So this one is better. It's actually in the lab report for the PCR part of this lab. But you probably won't have looked at that when you're ready to do the answers to the questions. So anyway, I'm going to post this for you. So let's talk about PCR for just a minute. PCR stands for the polymerase chain reaction. Polymerase is an enzyme that makes a polymer. Polymer means many subunits. That's all it means. Poly means many, mer means subunit, ace means an enzyme. So this is an enzyme that makes a big old molecule. That's all that means. Polymerase. DNA polymerase is the enzyme that makes new DNA. How does it do that? It puts new nucleotides into place after you unzip the DNA double helix so you can get to those bases that face each other. So the polymerase does that. It makes new pieces of DNA. Chain reaction, it just means that it happens over and over and over again and makes lots and lots of copies. So the basic premise of PCR is that you heat and cool and put all the proper ingredients in your tube along with a sample of DNA that you want to amplify. And the polymerase makes new pieces of DNA according to the little sections that you want copied. Well, how do you determine the little sections? You figure out what that sequence is that you want to look at, and you find primers. A primer is a little piece of RNA that you stick on either end of that section to get the whole process started. If you know anything about cutting grass and you've used a lawnmower before, you push the primer button, right, to put a little gasoline into your lawnmower engine to get the process started. And that's what a primer is. It gets it going. And then new DNA can be made. So you make lots and lots and lots of copies, billions of copies, in about 4 hours' time when you do PCR. Why have you heard of this before? Because this is one of the tests that is used to look for SARS-CoV-2, the COVID virus. You use the PCR, polymerase chain reaction test, to determine whether there's any of this RNA that's present in any of your nasal cells, up your nose, because you swab inside your nose. This is the most sensitive test that there is. Other tests test for antibodies in your bloodstream, and that means you've already been exposed to the virus long enough to the point where your immune system has reacted to it. That's the antibody test. But a PCR test is looking for the actual viral RNA, and you can do that. I know that I described that this is a way to copy DNA, but you can also copy any RNA there by adding a few extra steps. So here are the 3 basic steps to PCR. Denature. You have to break apart those 2 backbones of the double helix of the DNA because you got to get to those bases that are actually connected when the double helix is all together. So denature means to break apart the 2 strands of DNA. Anneal means to attach the primers to the target region of DNA that you want to copy. Amplify and extend, that's the 3rd part of the process, and that's where you make the new DNA because you've got the primers attached now. So you go through this. This takes about 3 minutes to do one of these steps. For the thermal cycler, that's the piece of equipment or the instrument that does this work, it takes about 3 minutes for one cycle to happen. Then it happens again, and then it happens again. So the thermal cycler does this over and over and over and over again. 30 to 35 cycles is usually what you run, and then you have this particular section of DNA that's been copied over and over and over again. So that is how PCR works. So you're looking for a particular target of DNA or RNA, and you're amplifying it or making copies of it over and over and over again, where then you can test for it, you can look for it. There's all kinds of things that you can do with it. You can use it. You can use it in genetic engineering if you want to put this particular gene into some other organism. So there are lots of reasons that you could do PCR. All right, one word about next week. So next week is week 6, and the lab experiment takes the longest time of any of the weeks for you to actually do. You're growing plants from seeds. Now, you're only growing the plants from seeds for 4 days, but you have 2 sets of them. So 4 plus 4 is 8. And one of those days you can actually cross over. So there's a minimum of 7 days of growth that you have to do with these sets of seeds. All right, so if you start next Sunday night, you are not going to be finished until the next Sunday night. I suggest that you start these plants growing from seeds by the end of this week, like Thursday. That's what I would do. I'm just making a suggestion, but just realize you're going to have to take pictures of these and show me that you grew them, and you've got to grow them for the length of time that we're saying. Otherwise, you can't tell which plants are dwarf plants and which plants are tall plants. There are 2 characteristics that you're looking for: the color of the stem, so either dark purple or green, yellow, white, so dark or light, and how tall the plant is. You'll have all your instructions in the lab, but what I'm suggesting to you is start as early as possible. My setup included, when I grew mine, you see that lamp right there? That's the lamp that I used to grow my seeds under. And, sorry about that. Make sure you use an LED light bulb. I mean, almost all light bulbs now are LEDs. Don't use a regular incandescent light bulb because that will burn up your seeds and your plants. But hardly anybody has those anymore. So anyway, you don't have to get a fancy light bulb or anything. Just get a cooler one, and LEDs are cooler. So with that being said, I hope that you have a wonderful week, and let me know if you need anything, you're having any problems, whatever. m.sigmon@snhu.edu. I'm here to help you. Have a great week.